Thank you, the organizers, for inviting me. So, to the science.
You should say where it ended.
Yeah, I got arrested. Hence I was deported from the event, but you should get it in the floor broadcasting. Just a reminder, I mean, Vicky showed us this already, but just so we are all in the same page. So this is the general principle of GPCR signaling. So there is the GPCR protein here in the membrane, and there would be a signaling molecule, maybe a hormone that would bind externally, and then the GPCR would activate the G protein internally. And that eventually, with a cascade of signaling, will lead to some kind of biological response, usually shutting down of genes or activating other genes. And I'm showing this slide because for this talk, what's important is that the directionality. So the GPCR has to be embedded in the membrane in the right topology, which in this case translates to N-terminal outside and C-terminal inside. And with that, the signaling would work properly. If it's the other way around, the signaling wouldn't work. So we noted that many GPCRs, they pair up. They form homo and heterodimers with other GPCRs. And then that there were also indications that beyond providing maybe stability, very often this interaction is important for the function. So as a means of amplification or control of the functionality. So we were wondering, since they have to pair up, maybe there are cases where the same protein chain includes both GPCRs, just linked to each other along the same chain. And we went to look for cases like that, and actually for completeness, we just looked for any case with more than one GPCR in the same chain. So that's a brief explanation of the procedure that we used. We just constructed hidden Markov models of all the 92 GPCR domains who were known at the time, eliminating trivial redundancy. And then we searched the database of protein, Uniprot 90, for cases with non-overlapping multiple GPCR domains. Then we clustered them and selected 16 clusters with at least 5 hits. The reason we insisted on at least 5 hits was because there's a lot of noise in the data, and we wanted to make sure that what we see is not due to just error in the data. So we assume if we see it at least 5 times, then it's very unlikely that the same mistake occur again and again. So it reduces the likelihood of us being wrong, which is always a problem with computation. So, and with that, we came up with 16 clusters and identical number of domains, and in total we had 57 proteins in those clusters, and then we mapped them to representative genomes. So this is a typical output of this effort. So you see in each of those mini panels, the x-axis is just the amino acid sequence of the protein, and the y-axis is the number of times that we see this GPCR in Uniprot. And typically we see, I mean, these are the interesting regions, like here and here. So we see a lot of times a stretch of maybe 2,000 amino acids, sometimes more, and that would correspond to a GPCR domain.
2,000 or 200?
200, sorry. Yeah, I'm a bit dyslexic. So this would be a GPCR domain, and here is another one, so this is a good candidate, and very often those would be large proteins that will have extra parts beyond the are sorted by the number of GPCR domains. And for example, you see this is our champion with 4 GPCR domains linked to each other. But this is something that we filtered out, and there are many examples. And you know that the problem with those computations, AI-based or otherwise, is that you get a lot of data and you want to reduce the possibility that you're making errors. So, for example, here, even though we see 2 peaks, and the first one is legitimate because it's 200 amino acids, but the next one is shorter, so it's just a fragmented GPCR. An interesting story by itself, probably, but for this talk, I'm not going to deal with that. And we actually still didn't study that. So we left such cases out. And then we put everything into AlphaFold. It was AlphaFold 2 at the time. And what you see here is the level of confidence in a way that you superimpose the predicted structure on the supposedly real structure in one residue, and you ask what is the likelihood of the rest of the residues to also superimpose well. So in each of those plots, here you have the sequence, and here you have the sequence, and you ask how well they superimpose, and it's color-coded, so the blue is very small deviation, meaning that here you have high confidence, and yellow is when you have no confidence or little confidence. So, and that's also an easy way to visualize the data, because obviously there is a very high confidence within the GPCR domain themselves, and so these are along the diagonal. So for example, here there is such case, and here there is such case. These 2, by the way, are too short, so we do not count them. So in this case, even though there are hints for maybe 4, we count it only as 2. Anyway, AlphaFold believes that it knows well where to predict the structure of the GPCRs, but then it basically tells us you feel free to orient them, like if you want you can orient them like this or anywhere. And of course it is not aware of not explicitly aware of the presence of the membrane which restraints. So in our case the dilemma is whether they're oriented this way or that way. Now AlphaFold does orient them with respect to each other, it does predict. And for the most part, it predicts them to be oriented in opposite topology. That is like this. So one of them would be signaling properly, and the other one would just be there, presumably not signaling, according to AlphaFold. But AlphaFold admits that it's uncertain about it. Okay. So the next thing we did was just to map them into various genomes. And you know, GPCRs, they are, so you can see here that on the x-axis are the 57 multi-GPCRs, putative multi-GPCRs that we predicted, and on the y-axis are the species. And you know, in general, GPCRs, they are present throughout the eukaryotes, from primitive yeast and so forth. But multi-GPCRs, at least where we detected them, they are not as broadly distributed. They are only distributed in chordata species from monkeys above down to reptiles and fish at the bottom here. You can see the images here. And these are the types of multi-GPCR that are there. We started with all known structures at the time, which included various types of GPCRs, and yet we did not discover multi-GPCRs with all of them. but only with those that are homologs to muscarinic acetylcholine receptor, adenosine receptor, and those that are listed here. So it's rather limited. And based on homology to existing ones, most of them are olfactory receptors, and some are chemokine receptors. You know, when you see a result, you try to rationalize it, and which we did. And in terms of chemokines, you can assume that, or you can rationalize that since smell is made of so many molecules, then you want something to be able to be sensitive to various types of small molecules. And if you have a multi-GPCR made of two or more GPCRs, then maybe one is sensitive to one molecule and the other to the other molecule, and then they signal accordingly, like collectively, or they don't signal if those two molecules are there. So you can imagine that this adds to some plasticity to biology, which would be nice. And likewise with chemokine receptor that should sort of smell the equivalent smelling molecules for the immune system, then again you want this complexity. You can think of it as evolutionary advantageous. Okay, so this is finally what a structure like this looks like. So I'm showing here a case with three GPCRs linked together in the same protein. We present them separately just because we are not sure how to orient them, but you can see them here. Three domain 1, domain 2, and domain 3. And indeed, we also mark the positively charged residues, lysines and arginines, because of the positive inside rule, Gunnar von Heine's rule, that states that generally proteins like to orient in the membrane so that positively charged residues are more abundant in the cytoplasmic side. And you can see that in both domains that there is a preference, and this is how we orient them. So we also show a superimposition of all of these domains on a beta-arrestin receptor with known structures. So this is shown in yellow, and the others are in different colors. And this is a complex. The reason we chose it is because it's a complex also with a G protein, and we can see the interaction region. And then when we rotate it, we see how it interacts with this specific helix that recognizes, and this is how it signals. There are specific motifs that are marked there in pink and red, and they facilitate the recognition. And reassuringly, in essentially all our 57 cases, each and every one of those domains, they feature those motifs. So that's an indication that they are functional, not only there. But I mean, there is this is an indication that there is evolutionary conservation of this region, which is important for the recognition and for the signaling. There are other motifs that are related to shift of conformation between active and inactive conformation of the GPCR, and those are also there. So again, reassuring that these are functional domains. Here is a particular case from wild yak, which combines chemokine receptor 2 and chemokine receptor 5 next to each other. And here we are pretty sure about the prediction, because there is essentially, because there's 100% coverage and very high sequence identity between what we see in our putative multi-GPCR and the known annotated receptor. So it's a combination of 2 and 4 chemokine receptors. Again, the structure of each of those receptors in combination with their chemokines. So you see the mark here. So in each case, the GPCR thing is in the relatively pale color, and the more bright color is the chemokine itself. And I'm showing this superimposed on the model of the multi-GPCR. So you'll see that this putative docking works reasonably well. It's another indication that maybe this thing really exists. So we went ahead into genome browsers and tried to look at it from a different angle, to see something about the expression and something about the organization of those regions in the genome browser, if it makes sense. So we can see here that so this is chemokine receptor 5, and this is chemokine receptor 2. This is in the cow genome, and you see first of all that they are close to each other. Only 5 kilobase pairs, which is considered a viable option for them to be co-translated, and also maybe in the same chain. Another thing is their order. So here you see that the chemokine receptor 5 is on the left, and the chemokine receptor 2 is on the right. This is conserved. So we looked at 30 different representative genomes, and in all of them, the order is the same. And again, I'm calling up evolutionary conservation argument that if the order wouldn't matter, then maybe it would flip. But not only that, also the relative orientation in terms of direction. So you can see that the direction is like this and like that, and this is also conserved among those 30 species. So again, probably there's some restraint for them to be oriented this way and to appear together, ready to either be a part of one chain or simply just being co-translated in separate chains. Okay, this is another scene for like a similar picture for human, and again you can see that everything is conserved, and the distance is 10 kilobase pairs. Now co-expression data for this particular pair. And you can see that in many cells they co-express, which is another indication for their importance to appear together. This is in human, and they are translated separately, but they co-express. So maybe sometimes they are just co-expressed as individual chains, and others they are along the same chain. Now the topology thing. So, as I said, for proper signaling, the GPCRs have to be oriented in the membrane in a certain way, with their N-terminal facing out and their C-terminal stretching into the cell. So, for proper signaling as a pair, both should be oriented this way. AlphaFold orients them typically like this, but is uncertain. So we took a close look at it, and again, that's the first thing I told you about the positive inside rule. So if we use it, then each of the domains individually should be oriented like this. But then there should be, because of the uneven number of helices, there should be a stretch that links from the C-terminal of this domain into the N-terminal of the other domain, which is above. So it should cross the membrane, and it should be hydrophobic enough for that, and long enough. And that's a problem. That's just a slide to show that otherwise there is high desolvation energy penalty, and it should be compensated with a stretch of hydrophobic amino acids. And our study, our calculation shows that for the most part that there isn't hydrophobic enough stretch. So this is actually the one case that we managed to find a hydrophobic stretch, and it's modeled like this, but for the most part there isn't any. So we come up with various very speculative suggestions as to how it works otherwise, without without this hydrophobic stretch. So one possibility is that the stretch really doesn't cross the membrane. There are 7 transmembrane helices in the N terminals. That would be like the simplest case with 2 GPCR. And here is the other one, again, 7 transmembrane helices, and they are just oriented in opposite topologies. And then maybe on Monday they would be oriented this way, and on Sunday they would be oriented this way. We don't know. The positive insight rule wouldn't dictate a preference in the orientation, because either one GPCRs will be unhappy or the other. And then sometimes when it's oriented this way, signaling will go this way, and sometimes it will go through the C terminal. 1 minute. Another possibility, and then the reason for this to happen is just to increase stability in the membrane. Another possibility is that there is truncation. So the first one goes right, and then there is truncation, and the second one just inserts independently, where the advantage is for them just to be together, next to each other, translated together, and ready to work together. Last possibility, and this is even more speculative, is that first insert into the cytoplasmic membrane, and the other one is inserted into the membrane of some kind of organelle. And that would contribute to making the organelles close to the membrane. And if that's a possibility, then it means that there is perhaps that direct signaling from the outer part of the cell directly inside this organelle, and vice versa. But it's yet to be detected. And finally, I want to point out a paper that Nikki Kozlov showed me recently that says that they examined splice variants of GPCR, and they saw that occasionally there are splice variants with 8 transmembrane helices, and with that, the N and C terminals are in the same region of the membrane, so the whole topology issue is nullified, if that's the case. And here is summary and outlook, but I'll skip it in the interest of time. And those are the people who did the project. It's mostly Kilic, a student from Turkey, who unfortunately after the war didn't make it back to Israel, so the project slows down, but hopefully it will continue. And everything was in tight collaboration with Rachel Kolodny from University of Haifa.
Having 2 olfactory receptors in the same cell, in the same transcript even, appears to be in violation of a rule that they have usually, and that is the rule of clonal exclusion, even allelic exclusion, where each olfactory receptor cell expresses only one olfactory receptor. That's usually the rule, and the rationale, at least in the mammals, and the rationale for that is that this could be a good way for the brain to tell what was the odorant by reducing it to the question, what was the neuron that binds it. If there is one-to-one correspondence between a neuron and a receptor, then all that the brain needs to do is to figure out which neuron fired, and it knows which receptor it was. So either that this rule is just violated, that's one possibility. Second, is that although these are two distinct olfactory receptors, maybe they bind the same odorant. Maybe their binding site is so similar, for example, that they bind the same odorant, and then the brain still has the simple task of asking itself which receptor bound. And maybe they don't bind the same odorant, but they bind a combination of odors that are typical of the same source. For example, both can come from a rotten fruit or from a dead body or fire or something like that. And then it really doesn't matter that they bind different odorants, but they come from the same kind of ecological or biological source. So have you looked into the sequences of these pairs or triplets to try to see if their binding site, presumed binding site, is similar or very distinct, or if we know whether they bind by their sequence identity?
We should, it's a very, very, very interesting point. Thank you very much. We looked at the binding site, but not as deeply as we should. In general, determining the kind of ligand that to bind is very, very difficult, because even as far as we saw, even GPCRs with very similar sequences, sometimes, at least according to the annotation, they would bind different odorants. So, we actually, within the context of another project, where we developed, where we are developing an AI-based tool, we want to develop the ability to detect suitable ligands for proteins, and when this one is done, which will hopefully, and if it's done well enough, we will use it here and hopefully be able to assign the odorants. Just in relation to your last point, I want to emphasize that the fact that we see this kind of pairing of GPCRs, and that it seems to be conserved in evolution, it means that it has some advantage. So it's not going to be a burden, like you say, that it doesn't matter because it comes from different, from the same source, different odorant. It probably adds some information. So maybe the fly would figure out that because there is a combination of two odorants from the same sheet, and this is deep sheet, and I should dive in, or something like that.
I totally agree. Totally agree. I'm just trying to see what could be the benefit.
Yes.
Okay, so let's turn on the